Today we shall discuss mineral nutrition. When soil is stirred in water, sand and silt will settle, but clay will remain in stable suspension because it is a colloid. It is small enough to remain in suspension and too large to go into true solution. The suspended clay particles will scatter light, causing the path traversed by the light beam to become visible. This light scattering phenomenon is known as the Tyndall effect, and this is a distinguishing feature of colloids and suspensions. A true solution such as sodium chloride or sucrose in water, on the other hand, will not scatter light. This is because in a true solution, the solute and solvent constitute a single phase. A colloidal suspension, on the other hand, is a two-phase system. It consists of a solid phase, the colloidal micelle, suspended in a liquid phase. Light scattered by the solid phase is responsible for the Tyndall effect. Compared to sand and silt, colloids provide an incredibly large surface area for interaction with mineral elements in the soil solution. The surface of colloidal clay is also with numerous negative charges, so it can bind and retain cations. This ability to retain cations and exchange cations on colloidal surfaces is the single most important property of soils insofar as plant nutrition is concerned. As shown in this example, the calcium cation is exchanged with the magnesium cation adhered to the soil surface. There are two types of colloids in the soil: the mineral colloid or the clay, and the organic colloid like humus. In the case of the mineral colloid, the simplest type is the kaolinite. The negative charges on the surface of the clay colloid arise by virtue of the ionization of the alumina and the silica at the edges of the clay particle. Inorganic colloid in the form of clay is not the only soil component that forms colloidal particles. Many soils also contain organic colloids called humus. Humus is an organic material that has been slowly but incompletely degraded to a colloidal dimension through the action of weathering and microorganisms. Because colloidal carbon is derived largely from lignin and carbohydrates, it also carries a negative charge because of the dissociation of hydrogen from the carboxylic acid, hydroxyl, and phenolic groups. Colloids like clay and humus are highly hydrated. That is, the positive pole of water is attracted to the negative charge surfaces of the colloid. The negative charge and the hydration contributes to the stability of colloidal suspension. The association of cations with negatively charged colloidal surfaces depends on electrostatic interactions. Hence, binding affinity varies according to the lyotropic series, reflecting the general rule that trivalents are retained more strongly than divalents and divalent ions more strongly than monovalent ions. However, ions are also hydrated, which means that they are surrounded by shells of water molecules, and electrostatic rules are modulated by the relative hydrated size of the ions. Since ions of smaller hydrated size can approach the colloidal surfaces more closely, they tend to be more tightly bound. Therefore, as we can see here, hydrogen ion, although it is a monovalent, electrostatic rules dictate that it is attracted more to the negatively charged surface of the soil than other divalents and monovalents. Cation adsorption is also reversible. This process of exchange between adsorbed ions and ions in the soil solution is known as ion exchange. The ease of removal or exchangeability of an ion is indicated by the reverse of the lyotropic series shown above. Thus, sodium ions are most readily exchanged in the series and aluminum ions the least. Consequently, any ion with a higher affinity to the negatively charged surface, for example, hydrogen ion, is capable of displacing an ion lower in the series, for example, calcium. The same is shown in this example, hydrogen displacing calcium ion, which is lower in the series. Alternatively, an ion with a lower affinity, for example, ammonium ion, by mass action can displace an ion of higher affinity. The immediate source of cations by the plants is found in the soil solution. As plants absorb soluble nutrients, they are continually replaced by those adsorbed in clay or humus. These ions are not easily lost when leached by water, and roots normally secrete hydrogen ions which assist in the uptake of nutrients. Eventually, the reservoir is replenished by ions derived from the weathering of rock particles. In this way, ion exchange at the colloidal surface plays a major role in providing a controlled release of nutrients to the plant. Cellular respiration in the root releases carbon dioxide to the soil solution, where CO2 reacts with water to form carbonic acid. Dissociation of this acid adds hydrogen to the soil, which help in the uptake of nutrients. Ion exchange may not always work to the advantage of plants. One effect of acid rain, for example, appears to be the displacement of cations from the colloidal reservoir due to the high concentrations of hydrogen ions. Those solubilized nutrient ions not immediately assimilated by the roots are readily leached out to the soil by the rain and percolating groundwater. Both the soil solution and the reservoir of nutrients are depleted more rapidly than they can be replenished, and the plants are deprived of an adequate nutrient supply. Anions are not attracted to the soil colloids, so they remain in soil solution. They tend to leach out of soil and find its way in streams and lakes, where it contributes to problems of eutrophication by stimulating growth of algae. The uptake of nutrients by most plants is enhanced by association of the roots with soil microorganisms, especially fungi. A root infected with a fungus is called a mycorrhiza. Mycelia is the body of a fungus made up of a mass of hyphae. Mycorrhiza are a form of mutualism, an association in which both partners derive benefit. The fine hyphae of mycorrhiza extend the reach of roots into the surrounding soil and facilitate the acquisition of mineral elements, particularly those of phosphorus, that are relatively immobile in the soil. In return, plants provide carbohydrates to the mycorrhiza. The nutritional status of the host plant is a key factor in extent of mycorrhizal association. Soils with which are moderately deficient in phosphorus promotes infection, while those soils with abundant nutrient suppress association or may shift from symbiotic to parasitic interaction. Soils with extreme deficiency suppress association. The beneficial role of mycorrhiza, particularly with respect to the uptake of phosphorus, appears to be related to the nutrient depletion zone that surrounds the root. This zone defines the limits of the soil from which the root is able to readily extract nutrient elements. Additional nutrients can be made available only by extension of the root, that is, by growth of the roots into new regions of the soil, or by diffusion of the nutrients from the bulk soil into the depletion zone. The extent of the depletion zone varies from one nutrient element to another depending on the solubility and mobility of the element in the soil solution. The depletion zone for nitrogen, for example, extends some distance from the root because nitrate is readily soluble and highly mobile. Phosphorus, on the other hand, is less soluble and relatively immobile in soils, and consequently, the depletion zone for phosphorus is correspondingly smaller. Mycorrhizal fungi assists in the uptake of phosphorus by extending their mycelia beyond the phosphorus depletion zone, thus improves phosphorus absorption. Apparently, mycorrhizal plants find it advantageous to expand their carbon resources supporting mycorrhizal growth as opposed to more extensive growth of the root system itself. Two major forms of mycorrhizae are known: ectotropic and endotropic. The ectotropic form, also known as ectomycorrhizae, are typically short, highly branched, and sheathed by a tightly interwoven mantle of fungal hyphae. The fungus also penetrates the intercellular or apoplastic space of the root cortex, forming an intercellular network called the Hartig net. Unlike the ectomycorrhizae, the hyphae of the endomycorrhizae develop extensively within cortical cells of the host roots. The most common type of endomycorrhiza is the vesicular arbuscular mycorrhiza, or the VAM. The hyphae of VAM grow between and into root cortical cells where they form highly branched tree-like structures called the arbuscules. Each branch of the arbuscule is surrounded by the plasma membrane of the host cell. Thus, while the hyphae do penetrate the host cell wall, they do not actually invade the protoplast. The arbuscule serves to increase contact surface area between the hyphae and the cell. Less frequently, the VAMs form large ellipsoid vesicles either between or within the host cells. The presence of the arbuscules and the vesicles provides a large surface area for exchange of nutrients between the host plant and the invading fungus. All organisms require nitrogen. Green plants obtain nitrogen in the form of nitrates and less readily as ammonium salts. Once assimilated, nitrate nitrogen is converted to organic nitrogen in the form of amino acids and other nitrogenous building blocks of proteins and other macromolecules. Nitrogen moves further up the food chain when animals consume the plants. Nitrogen is eventually returned to the soil through animal waste or the death and subsequent decomposition of all organisms. In the process of decomposition, organic nitrogen is converted to ammonia by a variety of microorganisms, including the fungi. This process is known as ammonification. Most of the ammonia is recycled to nitrate by soil bacteria. The first step in the formation of nitrate is the oxidation of ammonia to nitrite by bacteria of the genera Nitrosomonas or Nitrococcus. Nitrite is further oxidized to nitrate by members of the genus Nitrobacter. These two groups are known as nitrifying bacteria, and the result of their activities is called nitrification. The bulk of the atmosphere, 78% by volume, consists of molecular nitrogen, N2 or dinitrogen, an odorless, colorless gas. In spite of its abundance, however, higher plants are unable to convert dinitrogen into a biologically useful form. The two nitrogen atoms in dinitrogen are joined by an exceptionally stable band, and plants do not have the enzyme that will reduce this triple covalent band. Only certain prokaryote species, like bacteria, are able to carry out this important reaction. Both invasive and free-living nitrogen-fixing bacteria are the primary source of nitrogen for plants. The bulk of nitrogen fixed on a global scale is accounted for by the reduction of dinitrogen to ammonia by living organisms, among them the bacteria. Nitrogen fixation is a prokaryote domain because only prokaryote organisms have the enzyme complex called dinitrogenase that catalyzes the reduction of dinitrogen to ammonia. Prokaryotes that fix nitrogen, called nitrogen fixers, include both free-living bacteria and those that form symbiotic associations with plants, as mentioned a while ago. It is generally agreed that symbiotic nitrogen fixers, particularly legumes, contribute substantially more nitrogen to the soil pool than do free-living bacteria. The host plant provides energy in the form of photosynthetates and in turn receives a supply of nitrogen compounds for its own growth and development. The product of nitrogen fixation is ammonia, which is rapidly incorporated into amino acids. Plants that do not form nitrogen fixing associations generally take up nitrogen in the form of nitrate. Nitrate must first be reduced to ammonium before it can be incorporated into organic molecules.